Actually, I started it. All right, welcome everyone. Who's feeling great? Yes, who had an awesome summer? Nice, good, awesome. So welcome to the 2024 edition of our computational biology class. Today, we're going to be giving you an overview of the whole class. What are the modules? What are we going to be covering, etc. And then we're going to be starting with module 1 on Tuesday next week. So basically, lectures are going to be right here, right now. So if you're not here, you know, this information is not very helpful to you right now. But Tuesdays, Thursdays, 1 to 2:30. And then Fridays is recitation in the same room at 3 p.m. Everybody able to make it Friday at 3 p.m.? Raise your hands. Awesome. Good. Who's not able to make it on Friday at 3 p.m.? Okay, a couple of people. You know, we'll record everything, try to make it all available. So what are we going to be covering? We're going to be covering a whole range of both biological and computational topics. So we're going to be dealing with genomics and epigenomics, with the circuitry of human disease, understanding the three-dimensional structure of proteins, understanding immunology, imaging, ligand binding, single cell analysis, regulation and gene networks, and so on and so forth. So it's an enormous amount of material that we're going to be covering. And the best part of the class is what you're going to be doing with the material, namely all of the awesome final projects that are materializing every year. So every year, students who have never done any computational biology before come up with the most amazing, creative, forward-looking projects. And many of you become active practitioners in the field and just go off and write theses about this, write papers, publish these papers at conferences, and so on and so forth. So it's really just remarkable to see this extraordinary progress that happens every year. So this year, we have, again, revamped the class with not just genomics, as it has traditionally been, but also with 3D structure, with next generation GenAI methods for proteins and for chemistry. And that's where the second professor comes in. So I'm Manolis Kellis, and this is?
I'm Eric Hall.
So stand up, Eric, and tell us a little bit about yourself.
So I'm kind of like the, come up here. I have been known as like the microbiome guy or the poop guy, and that's kind of defined my career for the past 20 years or so. And I've known Manoli that entire time. We first met like right before I was applying for a job here. He's one of my favorite people in computational biology, and I had planned to teach a seminar class this term, which is very easy for a professor. You just like get students to like pick papers and read them, and you sit in the back and say, you know, make some comments. But when Manoli asked me, do you want to teach this class with me? I kind of jumped at the opportunity. I am transitioning from mostly doing microbiology to mostly doing generative AI. And so as part of that, last spring, I taught a very similar class, Gen AI and biology with Bonnie Berger. And that was part of my like introduction and like learning about the material. So I figured I could like reuse most of that material. Term, the way it's worked out, be like along the way, bear with me. The silver lining is that like I'm really excited about this stuff. I'm like learning it, you know, also along with you. So hopefully my enthusiasm or you know getting some of the material fresh. I've
Okay. And we are, oh, and I'm going to turn off the captions. Turn it away. And we know how to turn off the captions. There you go. Okay, awesome. So again, here we are. Hold on one second. Let me hide this as well. There you go. So basically, the goal is to cover both the foundations and the frontiers of computational biology, combining theory and practice, and then understand the principles of machine learning and algorithm design, understand influential problems and techniques, analyze large-scale biological data sets, apply this to human disease and to drug discovery, understand genomes with sequence analysis, regulatory motifs, epigenomes, understand gene expression and networks and RNA, understand evolution, comparative genomics, phylogenetics, understand genetic variation and disease circuitry, and then transition to understanding therapeutics, drug discovery, protein folding, and ligand binding. So basically, we're going to go both from the linear, the two-dimensional to the three-dimensional. And we're going to basically combine both classical machine learning and algorithm techniques like Gibbs sampling, expectation maximization, hidden Markov models, Bayesian inference, as well as the frontiers of generative AI and deep learning with sequence and graph-based models, geometric neural networks, deep generative models, and so on and so forth. Okay. So MLCB is machine learning in computational biology. So that's the list that you should be hopefully all on. And then dash staff is going to be us. And then we're going to be messaging you through Piazza, through Canvas, and all of these things. So basically, again, I'm going to try to spend relatively less time on course overview and sort of the logistics and admin trivia, and more time on why this is so ridiculously exciting and cool. Okay. So admin trivia. So you met Eric. So I'm Manolis. So I'm a professor in AI and computer science here at MIT. I'm also a member of the Broad Institute of MIT and Harvard. I'm a professor in both the computer science side of things and also the genomics, disease circuitry, epigenomics, and much more on the GenAI, protein structure, chemistry, graph neural networks, knowledge graphs, and so on and so forth. So we have 3 TAs with us. So Jared, do you want to quickly stand up and say hi?
Hello, everyone. I'm currently.
There are some seats in the front if you guys want to come. Yeah.
Yeah, I do research in the group in Department of Chemistry. Really excited about protein language models, and we'll have a recitation tomorrow at 3. We can go over
And Sarah?
Hi everyone, I'm Sarah. I'm a 5th year PhD student in ECS. My lab is Deborah Marks's lab at Harvard, and I work on using protein language models to predict viral evolution and for vaccine design.
Benjamin?
Hi, I'm Benjamin. I'm a PhD student in Manolis's lab. I do a lot of single cell disease dissection, epigenomics, regulatory genomics.
All right, so that's who we are basically. And there's also a survey out there so that we know who you are. So basically, there's a few folks who have already filled out the survey. It should only take less than 15 hours, I think. There's a lot of questions, I'm sorry. But a lot of them should be check. Who has done the survey? Okay, who thinks it was longer than you were expecting? Yeah, sorry. So who started and it was like, oh, gotta run to class? Yeah, exactly. So I apologize for that. I should have warned you. But anyway, it's going to be helpful for organizing both the fine resolution content of the lectures, but also for coordinating final projects, etc. So basically, do spend a moment to sort of, and it also helps, in my view, to sort of reflect on what are your goals for the class, because that's important, sort of a form of active learning where you kind of decide what you really want to know about. Okay, so again, we talked about all that. Course website, super easy, Comp Bio for computational biology, and then MLCB for machine learning computational biology. So you just go there, it redirects you to here, and so on and so forth. Oh, and I just added you all to this calendar, so MLCB 24 lectures. So hopefully all of the information will appear automatically on your Google Calendar. Okay, so what's our goal? Number 1, introduce you to computational biology. So basically both fundamental problems in computational biology algorithmic and machine learning techniques for data analysis more broadly. So a lot of the techniques that we're going to learn about today and throughout the semester are going to be applicable not just in CompBio, but in every applied data science, where you have to deal with the real world, with noisy data, with models, with approximation, with summarization, with building representations of the world. And I'll dive much more into what that means at the end of the lecture. And again, our goal is to not just expose you with stuff that was written back in 1905 when Einstein figured out how the world works, but stuff that we are still figuring out now and that we will know more about by the time the course ends than we did before the course started this semester. And some of which you will find out that we don't, and the world doesn't know about yet. So again, this is a very active, living, breathing field, and it's so exciting to sort of be teaching this year after year and to sort of see how the field is changing. And again, our goal is not to just to say, oh, download that program and then this program, make sure you set that parameter. No, that's not what this course is about. We are here to teach you how and why the methods work so that you can think algorithmically, you can think sort of from a machine learning and AI perspective, how to design the next generation of methods. That's what we're here to enable you, not just, hey, use these tools, but why and how to think about them. That's goal one. Goal two is the ability to tackle research. So we're going to be going through milestones for a final project that will allow you to build on the frontiers of the literature, usually with papers that were only published weeks ago, if not, in a year or two, because they're already on bioRxiv and you can kind of download the data. So for that, we're going to have both problem set questions that introduce you to labs with sort of real code from real papers that sort of push you to the frontiers to sort of be there where the papers are now. And some components of designing a part of an algorithm that you're going to have to then code up and sort of see how it runs, etc. So we're going to have some programming assignments, and the final project experience will allow you to both propose and carry out your independent research, and then present your findings both in a written form and an oral form. So we like to think about the two dualities of the class. So basically 4 axes, if you wish. On one hand, there's computation and biology. So basically, we're going to be learning about real biological problems, not biologically inspired algorithms. Like, we care about biology. We care about sort of solving biological, chemical, medicinal, problems. That's the first axis, biology. And the second axis is we don't just care to solve it with, just boring techniques. We care to sort of teach you fundamentals of computation and analysis and AI and machine learning and statistics and algorithms that you will then be able to apply in many places. So these are the two, first duality, computation and biology. And the second duality is foundations and frontiers. So basically, on one hand, there's the foundational stuff that, everybody should know. And then there's really sort of frontiers that might or might not become the next best thing. And we're going to try to introduce both. Typically, at the beginning of each module, we're going to be covering more frontiers and then, more foundations and then more frontiers as we go. So everyone should have a copy of this. If you don't, please raise your hand. We have a few more in the front. So what is this? This is basically the course at a glance. So you'll see that there's a lot of information compacted here. So let's walk through it briefly. So in the center, you have the blue alternating light and dark blue of the weeks. We're here in week 1. On week 2, we start the first module that's going to last across weeks 2, 3, and 4. And then second module, weeks 5, 6, and 7. Third module, weeks 8, 9, and 10. Every module comes with a problem set. The problem set is out as the module starts on Tuesday, and then it's due as the module ends on Monday before the next module begins. So every single time you hand in the previous problem set, and then you show up in class, we start a completely new topic, and you start a new thing. As opposed to, we teach you for 2 weeks, and then you start the P set, and you sort of are trying to figure out what happened 2 weeks ago as you're learning about new material, okay? So it's going to be a sort of coordinated homeworks along with the modules. Everybody with me here? Awesome. Second, we're going to have only 3 P sets for the 3 modules. And then module 4, we're not going to give any more P sets. The quiz is going to be right there. So, and it's only going to cover modules 1, 2, and 3. So any material here, you're not, quizzed on. Again, you're not here because you are forced to do it. You're here because hopefully you love to learn, and hopefully you'll care to learn about those as well. And some of you will be using them for your final projects. So basically anything that's covered in all of these modules is, of course, fair game for your final project, okay? So these are the homeworks, and then these are the modules. Now, notice there's a homework 0, and I messaged you about that in our announcement earlier today. So homework 0 is just to make sure that you know how to use all of the tools that we're going to be using. So Jupyter Notebooks, PyTorch, basically BioPython, Colab, and just the basic foundation tools that we're going to be needing for the rest of the PSET. So basically, for some of you, this will be trivial and you'll be like, why am I doing this? And for others, you'll be like, whoa, this makes no sense. Spend the time to learn it. Basically, we could have just required a bunch of other classes as prerequisites, but instead we're like, listen, if everybody really cares, then you're going to be doing it, on the first weekend. So basically tomorrow's recitation, we're going to go through PSET 0. So, and then we're going to talk a lot more about the projects, and we're going to be doing some brainstorming. So please try to fill out the survey before tomorrow, so that, when we get together at recitation, we can kind of talk about who's interested in what, form groups, etc. Okay. And then this column here is about the project milestones. Okay. So notice that at the end of every second week, there's a mentoring session. Okay. What is that? That's basically when we ask you to sort of complete little parts of the project. The first part is just simply fill out a form, not a form, but a one-pager that's like already like with some, entries there, which is a self-profile. Why? So that others can find you and you can form teams. Second, and also a little self-intro video. Why? So that you get used to recording yourself speaking publicly, because that's going to be part of your final presentation. We don't want this to be the first video that you record. Of course, you're not my age, so you guys have been recording yourself since you were babies. But some people are still shy. So basically, that's the self-introduction. Okay. Then second milestone at the end of week 4 is going to be selecting previous papers. That basically means find some cool papers about the areas of interest that you have that you could build on. And then if you choose 1 or 2 or 3 papers, we can kind of say, yeah, this one's great, this one's not so great. And we're going to be guiding you through that. Okay. Third milestone, or actually that's 0, that's first, that's second. Second milestone is project feasibility and proposal. So what does that mean? That means that you can actually download the data from the paper. We don't want you to sort of arrive here in week 13 and say, oh, it turns out I could only get the first 3 lines of this, massive database because the rest is behind a paywall. Okay. So feasibility basically means you have downloaded the code, you have downloaded some data, you have downloaded sort of all of the tools that you need, and you can kind of reproduce some version of figure 1. Does that make sense? So that's, feasibility. Then we're going to have 2 milestones after week 8 and week 10, which are sort of more like office hours. So you're going to be giving us updates. We're going to be giving you feedback. Okay. And then milestone 5 is important. Notice you don't have a PSET here. So you're going to be able to focus on that. So, and it's, a week after your quiz. So the goal here is to work backwards from the write-up to basically say, okay, here's what I had proposed. Here's where I am. Here's where I want to be. Let me make sure that all of the sections are there. Because again, something that we've seen is that by week 14 in the evening before it's due, you've done this amazing job in, AIM 1.1, and then you're like, oops, I still need like 1.2, 3, et cetera, to sort of complete. So that's the mid-course report. The goal is to sort of well before the project is actually due, to sort of have you reflect upon sort of where do I stand, et cetera. Okay. Then your write-up will be due on Friday before the end of the semester. And then on Tuesday, which will be our last class time, we're going to have you present in class. So basically, why are we doing this? So that people are not sort of staying up until 4 a.m. finishing their project, and then they show up and they're like, and they can't remember what their name is. Instead, we're going to have the project due on Friday. We're going to be a little flexible with extensions, but don't go wild. The slides due on Monday at midnight so that you get some sleep. And then the presentation will be live on Tuesday. Okay. Again, we're just here to help you. We're not trying to trick you or penalize you. Sounds good? Awesome. So that's basically the course. And now we're going to talk for the rest of the lecture today about the actual content. Okay. So on the right here, we have module 1, which is about genomics, epigenomics, single cell networks, and circuitry. Okay. This by itself could have been a fine 2-semester course. We're going to give it 3 weeks. Module 2 is going to be about protein structure, protein language models, geometric deep learning, and folding. And that again could have been a great semester, and you're going to get 2.5 weeks. Module 3 is going to be about chemistry and therapeutics and graph neural networks for representing the language of chemistry and docking and therapeutics. And I'm not going to make the same joke, but it still applies. It's not a joke, actually. And then module 4 is going to be everything else. Okay, so electronic health records, there's a great class on that one lecture. Imaging, there's a whole other class on that one lecture. Comparative genomics and evolution, both Eric and I used to teach whole classes on just those 2 lectures, and then metabolomics, etc. Okay, again, what's our goal? Our goal is to expose you to the awesomeness of the field. It's not to cover every last part of it. Okay, that's for you guys to just, once you have an entry point, go figure out the rest. Sounds good? Who's excited? Yay, awesome, good. All right, so if you want to know what this is going to be about, just go and watch some YouTube videos. All of the lectures from previous years are there. Some of them are on Panopto, some of them are on YouTube. Sounds great. For the in-class quiz, again, we call it a quiz. It's not a midterm, it's not a final exam. It's friendly, it's fun, it's fuzzy, it's cute. Your goal is to demonstrate the mastery of the materials in the 3 modules, not 4 modules. And to understand the key points emphasized in lecture, to understand the subtleties revealed in the P-sets, to tackle and apply new skills, to solve practical problems. We're going to have some true, false, and justify, some multiple choice, some short answers, and some practical problems, and maybe one design problem. Okay. The final project I mentioned, the goal is going to be through these milestones to frame a computational problem biologically. Biological problem computationally, to gather the relevant literature and data sets, to solve it using some new machine learning, new algorithms, and then to interpret the results biologically. So we want you to sort of wear both hats interchangeably, first the biologist, then the modeler, then the computer science guy or girl, and then the interpreter, and then the presenter. Okay, so basically you're going to be changing hats all the time. Okay. And our goal is to prepare you for the real world out there, where you're going to be applying for fellowships and grants. So your proposal is going to be drafted in a set of aims of what you're going to be accomplishing in the future. You're going to be working in teams, and that's extremely important. Again, learning these collaboration skills with people from very different backgrounds is extremely important and central to interdisciplinary fields like computational biology. We're going to be having some peer reviewing. We've done that in previous years. Folks loved it. We might do it again this year, of basically evaluating your peer proposals, where basically everyone is going to get 1 or 2 or 3 proposals, and then you're going to write feedback according to some criteria that we're going to provide, and then identify points of failure and suggest improvements. And then you're going to receive this feedback either from us or from your peers, or from both, ideally, and then revise your proposal, and then eventually write up your results in a scientific paper format, and then present a research talk to audience of your peers. Okay, so our goal is to mirror this process in 1 semester of what usually takes 3 or 4 years. Okay, sounds good? Good. When we asked you how much guidance would you like, usually everybody says the more the merrier for, guidance, how to start, how to continue, how to go on, etc. So basically that's why we have these milestones that I mentioned earlier. And the goal of these milestones is to ensure both sufficient planning and sufficient feedback. We don't want you to sort of start going down a track, build for weeks, and then come back and say, oh, actually, that's not even relevant, or that's not, feasible, and so on and so forth. Okay. So again, build a team of common interests and ideally complementary skills. I wouldn't want to see sort of 3 hardcore machine learning people or 3, purely medical folks, working, in separate teams. I'd love to sort of see people with complementary skills. And that's our goal. Our goal is for you to sort of find peers that will be your, partners, maybe not just through this, but maybe through your PhD or through your master's or through your undergrad. For inspiration, we gave you links to last year's projects and some recent papers. The goal of the proposal is to establish what will be your future milestones and deliverables and your expectations so that you can track your own project and, see how realistic were your views. Again, the mid-course is about seeing the endpoint, outlining your reports, writing already your methods, outlining what figures you'd like to have. And we're going to have these periodic mentoring sessions where we're going to team you up, not just with the TAs, but the TAs are going to help you, if you don't do it by yourself, find mentors who are practitioners in that field. So basically, among all of the folks here in this class, we represent many, many different departments, sometimes labs where you're doing, your Europes or your masters, your PhDs. And that will allow you to find mentors who can help you on the data sets, on the interpretation, et cetera. And that's part of, being a scientist, finding mentors and working with them. Okay. We also have partnered in the past with Communication Lab, which is really awesome and allows people to strengthen their writing skills, their presentation skills, et cetera. We have also engaged a personal private tutor for every one of you who is extraordinarily smart, is able to pass the bar exam, apparently, according to the news, and et cetera. So you know who I'm talking about, ChatGPT, our best friend. So ChatGPT is okay. We want you to work with ChatGPT. We want you to engage it, not just to, oh, please solve this for me, definitely not that, but instead, hey, help me think about that. Or, what does this mean, et cetera. So last year, I sat in the front as we had a guest lecture, and I noticed that the person next to me had just ChatGPT open and kept asking questions about, different words and different concepts. That's okay. We love you to do that. And you should push it to its limits. You should basically say, so that's our ChatGPT policy here. You should acknowledge it the way that you would a collaboration partner. And you should also save your chats because we might ask for them. If we basically say, well, it doesn't seem like you did anything, then just show us your chat and sort of, that'll help. And, we might ask you to submit it with your homework, but usually no. We just want you to be transparent. Again, our goal is not to get a bunch of homework done. We are putting the homework out there. We don't want it to get done. We want you to learn. Okay, so that's the only reason we're doing that. And if you use ChatGPT to enhance your own learning, that's amazing. Okay, so to summarize, we're going to basically have 25 lectures, each 1.5 hours, 4 to 7 meetings of mentoring, 3 P-sets, 30% of your grade. The final project is going to be 40% of your grade. Again, that's a huge chunk of your experience. The quiz is going to be 25% of your grade. And then the rest we reserve for participation and all kinds of other stuff. And again, collaboration is allowed with both humans and AI. And just acknowledge your collaborations, no outsourcing, no, hey, why don't you do this one? I'll do the next one. Definitely not that. Okay, any questions? Eric, anything to add, or Jared, or Sarah, or Benj? All right, so now the fun begins. Why computational biology? Why generative AI? So why is computation well suited to biology and genomes, et cetera? Okay, so that's a question that I've asked every year, and I'd love to hear your answers. So first of all, any questions? Can I get a 5, 4, 3, 2, 1 as to how well you're following? 5 is awesome. Yes? Good. Awesome. Great. Who does not have a right arm? Or a left one? Left side, okay, good. All right. So who's with me? 5, 4, 3, 2, 1? Awesome. Fantastic. Feel free to ask any questions at any point. That's the point. Okay. So why computational biology? Okay. So raise your hands. All answers are good.
I guess in physics you can use math because it's a lot more well-defined, but in biology it's very hard to actually use just the pure equations to model things. And model like complicated systems or complicated interactions.
I love it because you're asking a much more specific question, which is not just why equations, but why computation. And you're distinguishing math and computation, and you're saying, oh yeah, math, that's great for physics because you have an equation and then it describes the process. But with biology, I want to rephrase what you're saying by saying biology is hacky. it didn't get there through one beautiful equation that, I don't know, Einstein or God or someone wrote, and then the world followed. It got there like an engineer, like tinkering and, messing with stuff and trying different solutions. So to understand biology, you need something different than just mathematics. So that's a great answer. Other answers? Yeah.
Very much said. Many scientists want to ask many different questions about it and want to process it.
I love it. I love it. So big data, multiple questions, iteration. Love it. Yeah.
The kind of data that you usually get from computational biology is like sequences, which is really hard to understand.
Good. So the fact that machines can actually understand the language of biology better than humans. We did not invent the first programming language. We inherited the first programming language. So DNA is a programming language, and it's written in a language and in a mindset that is not very well fitted with the language of humans. But again, we can debate that over evening conversations. Because one could argue that maybe the human brain evolved from the same type of design principles of decentralized control and, emergent behaviors as perhaps gene regulation itself, and so on and so forth. But I really like that. It's not a human language. It's a language of something very foreign. Great answer.
More like a generative AI. I spent a lot of biology is about generation from DNA to RNA to protein and how things are constructed.
I love that you're starting from a code and you're producing stuff from that code. So you're instantiating gene transcripts and you're instantiating protein folds and you're instantiating protein-protein interactions and chemical protein interactions and so on and so forth. And we can model with simulation or with machine learning, as we're going to see, these types of processes in the same way perhaps the cell is doing that. And what I like about what you just said is that when you look at image interpretation from machines and image interpretation from humans, we tend to make the same mistakes. It's kind of very interesting. It basically says that there's something fundamental about the interpretation process, and maybe the misfolding of a protein in actual cells will be similar to the misfolding of the protein in our computational engines. Very cool. Yeah.
Biology is very time intensive and computation can help hypothesis.
Good. I have the choice between, hey, why don't you go do this experiment and tell me in 6 months if it worked, or hey, why don't I run 1,000 of those and then I can see which one's more likely to succeed. And yes, it's an approximation, but it can save me a lot of time because of the very different timescales of experimental versus biological sciences. Yes. Can be more. Oh, that's so interesting. It's not just that I don't want to wait for the experiment, but I want to actually do the experiment because maybe I care about animals. Maybe I care about, human tissues, or definitely don't want to experiment in humans, and so on and so forth. So yeah, and also when you're thinking about a medical intervention, for example, should we, in a population where there's an outbreak of a disease, should we impose a particular, solution? Maybe we should run a simulation as to what might happen before doing that. So great point. I like that.
Things can actually inspire people to find some biological questions that they have never been think about.
Good.
And so
Good.
it's people now intuitively to people that mainly ask questions based on their experience. But our experience is very rich, but it's limited. So sometimes maybe use the computational methods to actually analyze the data and to let us to some new things.
Yeah. Let me describe this as getting out of our local maxima. Perhaps we can kind of use computation to explore ideas and hypotheses that we as humans are kind of maybe not really amenable to right now, because it will give us a vantage point from which we can kind of see further. And now that becomes appealing hypothesis. So it's really great, sort of basically pushing us out of our preconceptions and basically taking an unbiased data-driven approach to understanding biology. It's great. Next. Yeah.
towards biological data, sometimes we like make mistakes or there are a lot of flaws in it, but it takes recognition of like the higher level patterns in the actual biological system for us to fill in those gaps that we like created.
Yeah, it's a fantastic point. So what you're distinguishing is two types of errors, if you wish. The first type is when the cell makes an error, where instead of translating one amino acid, it translates a different amino acid because it misread the code. And that happens all the time. And you basically do a bunch of measurements, and some of them are just off because they're actually off. Another type is when the measurement itself is erroneous. So basically, there's a biological variability, and then there's technical variability. And what you're saying is that by building models of the world, we can kind of somehow find not just a reduced size representation, a compressed representation, but maybe a more robust representation, because that compression comes with the benefit of summarization, abstraction, and therefore perhaps ignoring the noisy, either biological or technical variation. That's beautiful. Other comments? Who thinks these were amazing? Right? 5, 4, 3, 2, 1. I'm blown away. Every year gets better. So you know, yay for human evolution. All right. So these are answers from last year. So basically, that there are rules, that it's all about finding patterns. It's all about data. There's lots of data and lots of interactions of these data. So it's not just lots of n, but also n squared and cube, etc. The ability to visualize, that's an important component. So basically, not just simulate and get the answer, but visualize, because engaging our visual perception system, which has evolved, frankly, for way longer than our, I don't know, tabular interpretations, or reading through ACGTs, is extremely important. So basically, I usually like to say rule number 1 of genomics is look at and there's 4 chairs in the front. Rule number 1 of genomics is like, look at the darn data, basically, just like, visualize it, try to sort of, engage your, visual thinking apparatus. Then, the ability to run simulations, these temporal relationships, being able to guess and verify, generate lots of hypotheses and then test them, propose a mechanism or a theory to explain the observations, again, building networks and combinations of variables, being more efficient. There's a lot of informatics infrastructure as well, the ability to combine data sets together from disparate places and sort of, get insights that would otherwise not be possible. Understanding, again, not just correlations, but also higher order relationships. And then the cycle of between hypothesis generation and testing, getting dramatically condensed. And the last one is my favorite, that life itself is digital. That basically, as somebody mentioned, that it is a generative process itself. That basically there's a code, there's some instruction sets, there's, interpretation that happens by the cell itself. So it's a very natural medium for which we can kind of, apply our own techniques. Okay, so great. This is basically a snippet of an actual genome. This is, this is basically what got me into computational biology. Probably longer than many of you have been alive. So that was back in 1999, so 25 years ago, where I basically first saw a text file of the human genome as it was being assembled. And I was like, oh my God, sign me up. So that was 99. The genome did not come out until many, many months later. But I saw this and I was like, oh, that's alive. That's my own code. This is what runs, inside my cells. So I was blown away. And as you start thinking about what's in these signals, you basically see that there's some patterns. For example, every protein coding gene starts with an ATG and ends with either TAA or TGA or TAG. So there's a start codon and a stop codon, another start codon here. And then there's motifs. There's like this Tata motif that you can see in front of many genes. There's these CGG spaced bilevonucleotides of CCG, CGG spaced bilevonucleotides of CCG, etc., which basically are binding sites for different proteins that then will turn genes on and off. So it's kind of cool. There's like a code out there and there's signals sprinkled in. And some encode proteins, and others you can think of as the red and lights that basically turn genes on and off. Okay, so these are just the most basic building blocks of genomes. So we could say, okay, great, genes start with an ATG, super easy. Let's just look for the ATGs, and these are the ATGs. So there's a signal to noise problem that we have. So basically, how do we extract signal from the noise? Okay, and that's where modeling comes in. That's where AI comes in. That's where machine learning and statistics come in. So basically, in addition to these motifs and these protein coding segments, the genome has a whole host of building blocks. So there's something known as enhancers, which are collections of motifs that act together to control where and when a gene is expressed. There's promoters, which are, again, collections of different motifs, which are, and again, motifs, in case it's not clear, are these things here. So basically, CGG11CCG or CCCCA, which in reverse complement is TGGGG, which is here. These are motifs. These are patterns that are recognized by specialized proteins that bind the DNA. Okay. Now, you might ask, how is it reading the DNA? Is it like opening it up and reading the bases? The answer is no. Every base pair, for example, an AT or a TA or a CG or a GC, every base pair has some atoms sticking around on the side. So these proteins feel the atoms on the side. So basically you have, there's a major group and a minor group. So again, having a molecule that has a major groove is kind of cool. Very groovy molecule, but majorly groovy. So basically, as you see that the stack of the ladder, the side of the ladder is actually legible, because if you have a C or G or a G or C, they will have different atoms on the side that you'll be able to feel. And that's basically how these motifs are recognized, okay? So if at any point the words don't make sense, please stop me. But, these are some building blocks. So basically, these are the motifs, and then these are the proteins. So the proteins are basically what, the genes translate to proteins, basically, and they're the effectors. And then the motifs are sort of the control regions. So basically, we're going to be talking about the genome, the genes, the control regions, the words of gene regulation. So these are the motifs. These are, then the regulators are the proteins that recognize these motifs. And of course, we're going to be talking about dynamics and networks and grammars. And then, how do the proteins actually fold? How do they interact with chemicals? How do you design therapeutics to match these structures? How do things evolve? How do they vary? And how do we identify those genetic associations with that link genetic variation with phenotypic variation, and ultimately understanding disease? So that's going to be a lot of our menu for this semester. Okay. So I added this section this year because I've been saying about computation in broad terms, and you guys answered spot on. But there's something different this time, something fundamentally different, and that's why Eric is basically saying I am so excited about this. And I think this time it's truly, truly different. And this is about generative AI, about deep learning, and specifically representation learning, okay? If it doesn't make sense yet, hopefully in 10 minutes, you'll see why my mind has been blown by what's happening. I think what's happening is that we are for the first time designing machines that can't just find patterns, but they can find meaning in biology, that they can truly understand the language of protein folding, the language of chemistry, the language of genomes, the language of gene regulation, the language of physiology, the language of disease and health. And I think that is fundamentally different. And I put together a few slides to try to explain this concept of representation learning, which I think is basically what generative AI is all about. I've heard many prominent scientists, amazingly smart people, give talks saying, oh, AI has been around since the 60s. The only thing that changed is we have more computation and we have more data and we have bigger machines. And I call bullshit on that. I'm sorry. There's something fundamentally different about representation learning. And I want you to get out of this lecture saying, oh, something really did change this time. Because AI has been around for a long time, but this time, it's really different, okay? Let me try to explain. So I gave this talk in Greece, so of course I put picture from ancient Greece here. This is from Epidaurus in 4th century BC. You basically have the, it's not clear if this was a god or some temple physician. The distinction was very subtle in ancient Greece, and I think it still is very subtle in Harvard hospitals. And you have some kind of priestesses or nurses that translate the words of the gods. And then you have a committee. This is peer review nowadays. And, you have a patient. So, maybe this hasn't changed that much. But what has changed is that the doctor is not just looking from the outside anymore. Okay. What we can do now is engage microscopes. You can basically peek further and further and further. And this is basically what changed in the Renaissance. Basically, the church finally allowed people to open up bodies. Actually didn't necessarily allow, but anyway. And then we have anatomy, we have microscopy. We're able to sort of start seeing inside the brain, inside, like we are studying these cells in my lab right now, and sort of having this dramatic transition from the body from the outside to the body from the inside was, the first fundamentally different thing. Now, if you look at Alzheimer's disease, it was diagnosed by Alois Alzheimer back in 1911, 105 years or 15 years ago. And the phenotypes, could somebody close the door? The phenotypes that Alzheimer recognized were basically number one, amyloid plaques, and number two, neurofibrillary tangles, okay? How? By looking in the microscope. And you basically now have two variables. Are there plaques or not? And are there tangles or not? Okay. Now, those two variables are exactly what's being addressed by the drugs right now against Alzheimer's. They're just trying to remove plaques. It's not actually clear that they will actually function for Alzheimer's disease. But a lot has happened since then. The first thing that happened is genetics, being able to look at nucleotide resolution at what are all of the genetic differences associated with disease. Why is that exciting? Because it gives us causality. It basically tells us that a genetic difference here that I inherited from my parents correlates with whether I have Alzheimer's, which might also correlate with all kinds of other stuff. But correlation is not causation unless there's temporality or, of course, genetics, which gives you temporality, because I inherited this variant well before I chose to, binge drink every Friday. Okay? So that causality is extremely important and extremely high resolution. This is both a blessing and a curse. The blessing of genetics is that it doesn't matter how something works. You can find a genetic association saying this letter here has something to do with Alzheimer's. The curse is that you have this letter, you have Alzheimer's, you have no idea how. And that's what we're going to learn about in at least the first module of the class. How do we interpret genetic differences in the context of everything we know about biology to be able to understand the mechanism through which this causal variant is happening? Because if I understand the causality exists and the path through which causality acts, then I know where to intervene to reverse the disease phenotype. Is everybody with me here? Good. First change is, of course, microscope, causality. And the third one is massive amounts of data, okay? There's a 100-year difference between this picture and that picture. This picture shows 4 cells, 2 variables. This picture shows 2.4 million cells, 20,000 variables. Okay? Every dot here is 1 cell. And that 1 cell has the expression of 20,000 genes. So it's a 20,000 dimensional space that I've condensed here into 2 dimensions. And it is 2.4 million data points across 430 donors, half with Alzheimer's, half without Alzheimer's. Okay? So that's where AI comes in. That's where massive, massive, massive information needs to be processed. So that's the 3 shifts that I want to talk about. The first is we used to be all about hypothesis-driven research, and we're now all about data-driven research. So we could formulate the hypothesis, gather some data. You would do lots of thinking before, and then you have a targeted study that only measures one thing. And then the problem with that was it was highly biased and very little novelty. Today, we basically shoot first, ask questions later. So we gather tons of data, and then we have systematic data sets. We can build resources, massive amounts of data sharing, comprehensive use of biology, and then the questions and the models, et cetera, come up. That's the first paradigm shift. The second one is what I mentioned earlier, correlation More coffee, better health. More chocolate, more Nobel Prizes. Epidemiology has been about correlations. If I buy a $7 cup of Starbucks every morning, I have better health outcomes. Is it that Starbucks is so awesome? Or is that if I can afford Starbucks every day, I can also afford a better healthcare, I can go running, I have more free time, I have better food, nutrition, etc. Right? So correlation, not necessarily causation. Whereas with genetics that correlate with disease outcomes, then we know it's a unidirectional arrow. So we can create polygenic scores, understand causal factors, and with biology, unlike sort of the complicated ethics of intervening at the societal level, with biology, you can just do perturbation experiment. You can take some cells and poke things and then see what happens. So you can confirm the causality. And the third one, which is sort of what this section is about, is that we go from classical data analysis, which is the first part of why computational biology, where you basically build a new methodology for every problem. The human scientist does all the thinking, and there's very few parameters. We're trying to minimize the number of parameters. We have very targeted models about the question at hand. To generative AI and deep learning. And this is about building foundation models. Who has heard the word foundation models before? Raise your hands. Awesome. Who has not? Okay. So a foundation model is a model that is basically built on massive amounts of data, typically self-supervised, and has some emergent capabilities that were not inherently programmed in there. And that's what's spooky about this time it's different. That's what blows my mind. The fact that you can take massive amounts of data and just say, you know what? I'm just going to hide this part of the data. I'm going to have you predict it. And then to get good at predicting the data that I hid, which is basically a self-supervised approach. I didn't have any human labelers come and look at the data. I just said, predict the missing piece, randomly hide a small part of the data. That allows the machine to now get so good at predicting that part that to get good at predicting, say, the next word in an English language, you first have to understand word frequencies. And then you get, I don't know, to 80%. Then you need to understand the topic. Maybe you get to 83%. Then you need to understand emotions. And then you get to emotions, then you get to style, then you get to all kinds of subtleties about the human psyche. And then you get to 95%. And then the last 5%, that's where you build an even more complex model. And then it understands stuff that we don't know about ourselves. Okay. And that's what's foundational. That's what's extraordinary about this foundation model, that they have these emergent capabilities. Okay. That they're multimodal, they can understand data from multiple different modalities. And a lot of the projects that I would love to see, that I already saw in some of your suggestions, is what if I want to connect protein structure and protein function? How do I represent protein function? Maybe through a knowledge graph, maybe through the biological description in English of what this protein does in, the protein data bank, okay? What if I could connect the 3D representation of a protein with the English description of what that protein does? One I got through experiments, the other one is just reality. It's like, biology itself. If I can blend the two, the same way that ChatGPT speaks images and text at the same time. You can basically say, draw a picture of, I don't know, a bear riding a bicycle through the moon of Jupiter or something. And it does that perfectly fine. So why? Because it has learned a multimodal foundation model that understands images from the pixels up through this deep representation learning, and understands language from the bottom up, from every phoneme and every letter and every word to sentences and emotions and, the human psyche, okay? And by blending these two spaces together, it's able to speak both languages. We are right now in my lab building a foundation model that speaks protein folding and protein function at the same time. We're able to speak to it, and it will design proteins or interpret folds and sort of translate for us between the English language and those things. I want you guys to do the same thing. Just go and learn about protein, about patient trajectories, both the doctor notes and the structured data. and learn how to translate between the two, how to talk to your systems. I want you to sort of take on, I don't know, chemical structures and biological descriptions of what they do, or knowledge graphs and, you know, structure, or you name it. Like basically, I think that the time is there for us to truly push the frontiers of human understanding of biology. And I think that this generation of AI can actually do that. How? Through multimodality, through foundation models, the self-supervised learning and emergent capabilities, through representation learning, and through hierarchical concepts. And that's the deep part. The fact that you're learning a representation here, you're building on it to learn the next layer representation, the next layer representation. And that's what we call deep learning, okay? To truly understand concepts and get new insights. Do you guys see why I'm excited now? This is fundamentally different. Okay, so that's our goal. Basically, what we need to understand is how are disease-associated causal factors acting through the RNA, the epigenome, the networks, the circuitry of the cell. And that's going to be module 1, to basically understand how that eventually leads to disease. And we're going to be learning about these networks in module 1. We're going to be learning about regulators, the motifs that they bind, the enhancers through which they act, the genes that they target, the genetic variants that perturb them, the cell types where they become active, and so on and so forth. Okay, so that's basically module 1. And then that will allow us to now start intervening, basically to sort of say, okay, here's the strongest association with obesity. We understand how it works. We can go and intervene. And this work that we did in my lab to basically show that I can flip the switch of obesity back and forth and sort of turn lean cells into obese cells and obese cells into lean cells by changing different aspects of that circuit, okay? And we can do the same thing in cancer. We can do the same thing in Alzheimer's. We can do the same thing in many places. And then the next part is how do we now understand that structure of chemistry, of protein folding, and all of that, again, through these representations. So what do we mean by representation learning? When an AI understands this image and says it's a person carrying a backpack, okay? What does it do? It starts out with pixels, just like our retina. So basically we have pixels, picture elements, pixels, projected in the back of our retina. And then that's captured by neurons that actually learn how to recognize, based on the patterns of activation of those pixels relative to each other, learn how to recognize horizontal edges and vertical edges and shifts in color. That's the first layer representation. So you're going from pixels to representations. And then from those representations, I then build the next layer up, which is corners and circles and, you know, ellipses and you name it. And the next layer up is like, oh, okay, now that I have these basic shapes, I can sort of figure out what's an eye and what's a, you know, wheel and what's a nose and an ear and so on and so forth. And then I can build additional representation on top of that and eventually get to say, aha, that's a person. Why? Because inside either the machine's brain or the human brain, and I see a lot of parallels, so, you know, I'll be using them interchangeably. I have neurons that are activated by the previous layer to which they're connected, showing particular patterns. And then the next layer, again, builds on the patterns of this layer and so on and so forth. And what's fundamentally different about modern machine learning, modern generative AI, is that these patterns are not hard-coded. We didn't just bring a bunch of super smart engineers with classes and said, oh, okay, you should do this and you should do that, okay? We basically just let the machine figure it out by itself. We basically just like let the machine loose and it figured out what are, you know, the representations that I need at the lowest level to eventually distinguish cars, people, and animals. Can I get a 5, 4, 3, 2, 1 here? Who's with me? Okay, awesome. Who thinks this is like awesomely exciting? Awesome, good. So now that's the key concept, in my view, behind why this time it's different. It's not more data, it's not more computation, it's not bigger machines. Yes, of course, it's all that, but it's also fundamentally new algorithms. And the way to think about this is that here you have the y, here you have the x and y, okay? That's how machine learning has always been. You have a bunch of data x and you have a prediction output y. The why it's different this time is the z in the middle. The z is the latent space, the latent dimensionality, the embedding representation of all of these different variables. So if you look at the papers that changed the world in the world of image vision and that led the change in all of generative AI, it's exactly this. On one hand, you start by learning representations, and that's the modern part, the modern deep learning, where you're basically learning these filters, these patterns, these building blocks through convolutional operations, where you basically have, you know, an X here is basically a pattern that you're scanning through your image and you're saying, oh, I have a match, a match, a match, a match. And that's layer one. And then in layer two, you have another pattern like this, and you're like, oh, a match. That's building on the X's, and so on and so forth, okay? So that's the representational learning. And then, oh, sure, you can put the stuff that we had in the 60s, and that's a fully connected network. And the fully connected network is just, you know, approximation of functions. It basically just like, you know, does some kind of nonlinear nonlinear equation, nonlinear computation, basically. Okay? This part, yes, we had it in the 60s. But this part here, that's fundamentally new. Okay? And in deep learning, the two tasks are typically coupled. You basically start with an X and a Y, and you get better at predicting the Y. That's a human label. And then you get better at the Z, at the latent representation. However, the place that it gets really exciting is when you basically ignore the Y and just say, oh, I'm just going to hide a little patch of that image. I'm going to ask the machine to repredict it. Why is that exciting? Because then you can just like learn all day. You don't need any humans in the loop. You can just like, you know, let the machine loose. Just walk around and predict the next frame, or listen to human language and just predict the next word. And then to get good at that, you need to build their underlying representations to understand all of, you know, biology, all of chemistry, all of, you know, patients, and such. Okay, good. So then that principle applies to convolutional neural networks that you can basically use to learn images. You can apply the same principle to learn patterns in the genome, where you basically look for repeated motifs, basically, through this one-hot encoding ACGT representation of a genome sequence. If you have your genome sequence, you can turn it into an image. One dimension is only 4 pixels long, and it has ACGT. And now you look for repeating patterns in this 2D image, okay? You can do the same thing with graphs. So basically, we're going to talk about chemistry. Now, what is chemistry? It's basically just a series of graphs. Now, that's not the traditional lattice of a 2D image. That's not the one-dimensional sequence of a genome. This is connected in funny ways. And that connection allows you to now learn representations on graphs. How is that possible? What you want to do is at every layer, you want to build the next abstraction based on the previous one. So maybe if I'm trying to understand the function of a chemical, I start with a chemical, and I know that this chemical, I don't know, makes me itch or like cures, I don't know, E. coli infections, okay? So I want to sort of predict what is the output of this input. That's the X. The Y is itch or E. coli dead. And then the question is, what's the Z? What's the representation? Is everybody with me here? Good. So what's the Z? The Z is basically something that will allow me to learn from the bottom up. The same way that I learn with pixels as the primitives, I now want to learn with atoms as the primitives. And I want to start with one atom at a time. And that's my layer one representation. What are the properties of that atom? It's hydrophobic, it's charged, it's polar, and you name it, right? That's layer 0. Layer 1 above wants to use something more abstract. How am I going to do that? By asking, how is that atom placed inside the molecule? What are the other properties of the other atoms surrounding it? Okay, and that gives me the layer 1 abstraction. by saying, oh, this is a charged atom surrounded by two polar atoms, for example. And that now becomes a new representation. And then I can keep going up the hierarchy by basically learning initially only about my neighbors and then about the neighbors of my neighbors, because I'm now using the layer 1 representations of my neighbors to basically build my layer 2 representations and so on and so forth. And then I can use that to eventually predict something about that molecule. Is everybody with me here? So that's basically taking the same principle of bottom-up learning and representation learning to now learn the latent representation of chemicals. And I want you guys to do a final project that basically says, oh, I'm going to use the latent representation of chemicals and correlate it with, I don't know, the diseases where this chemical acts, right? You can do that, and you can visualize those, and you can sort of play with those, etc. The same thing applies to language. At the bottom, every word has some ground level representation. What is that ground level representation? I can learn it by learning how to predict a missing word in a sentence. And by predicting a missing word in a sentence, if that word is president, and I'm trying to predict the word president, the context that predicts the word president is actually kind of similar to the context that predicts the word king, or God, or leader, or you name it, okay? So that context will be similar for words that have similar meanings. Is everybody with me here? So that's the basic idea behind word2vec. The fact that I can take a word and represent it as a vector in space. And that allows me to now cluster words together that have similar meanings just based on that very simple approach. And then it becomes much cooler because I don't have to treat every word in isolation. The same way that I took chemicals and I looked at the atoms surrounding every atom and I had a new refined representation for that atom based on the neighborhood, I can do the same to go from a word to the meaning of a sentence or of a whole document. And that's where attention comes in. That's where transformers comes in. And that's where this whole idea of reshaping the latent embedding representation of a word. So this is the attention that this transformer model learns from just running it on a sentence. And now you can transform the word apple in a sentence from a basket of fruit to the apple on my desk, or sorry, next, like, you know, instead of using Windows, I'm using Apple, then suddenly the context completely changes. And that's the whole concept of going from the low level representation of one thing at a time to the high level representation of a whole sentence, okay? So, and again, the multimodality comes from the fact that I can go back and forth between images and text. I actually typed this to ChatGPT and it came up with that. And then I searched the web for an image and I asked it, what do you see? And it showed me that. But I mean, that's spooky enough. But now try taking the most complex figure from any of my ridiculously charged papers and just paste it to ChatGPT and say, what do you see here? It'll just go on and on about what's there. And that's the whole concept of multimodal representation learning. The fact that if you have a web's worth of images and their captions, you can then learn joint multimodal embeddings of cars and their descriptions, or, you know, dogs and, you know, their breeds and so on and so forth. And that allows you to now go back and forth where a point in this joint multidimensional space has both a language projection and an image projection. And this applies to proteins and chemistry and, you know, gene ontology and, you know, gene expression patterns, etc. Everybody with me here? So go wild with that in your final project, okay? So I want to make a small parenthesis that this Z vector, we could just simply say, ooh, AI is like Apollo in, you know, ancient Delphi, and only Pythia is allowed to speak with Apollo. Or we could say, you know what, that's our machine. Why don't we just take these latent Z vectors and just like play with them, visualize them, represent them, right? So we did this. We basically built a little team with a little group within my team that basically represents now these latent embeddings in an interactive, you know, playground, where here we took the 155,000 papers that have cited our work in our research group. And we basically projected every single one of them to a latent embedding space by sort of, having a large language model that reads these papers and then just ask it, hey, what's your latent representation? It's a 1,500 dimensional latent representation, but we can project it using methods that we're going to see later in the semester into 2 dimensions using t-SNE or UMAP that we're going to teach you about. And then you can basically see how are these clustering together in a Z vector space. I want you to do the same with chemistry. Just take every chemical and just cluster them and visualize them. Take every protein, cluster and visualize it. Take every gene expression pattern, cluster, visualize it, etc. Okay. Rule number 1 of genomics: look at the darn data, but don't look at the X space. Don't look at the Y space. Look at the Z space, and just have a blast. Everybody with me here? And by learning multimodal representations of any kind of crazy data set and language, then the Z vector will be interpretable because you'll be able to ask it about the language projection of that latent space. Okay? So you can do this with pathological maps of hundreds of patients. So basically here, every dot is a patient with Alzheimer's disease. And this axis turns out to be pathology. Basically, do they have plaques? Do they have tangles? This axis is cognition, and it's quite orthogonal. And you can ask, what are the genes that are expressed differentially along this axis and along that axis? And you're finding that along that axis, the genes that correlate with the variation in cognition are basically expressed in excitatory neurons. The genes that covary with pathology are in fact expressed in microglia and oligodendrocytes. This is kind of cool. It basically says that by learning these multimodal representations of patients and gene expression, we can basically start, peeking into some new insights. This is now taking this map that I showed you earlier of 20,000 measurements for every cell and 2.4 million cells across 400 individuals, and then projecting onto this map the phenotypes of those people, where you can basically say, well, who has Alzheimer's and who has, and who's a control. And based on that, you can now see neighborhoods of this transcriptional state that correlate with phenotypic variables. So suddenly you can kind of see that maybe the cells of these people can be pushed here to make them healthier or something like that. And then you can start understanding about the regulatory circuitry of how I can push them there. You can do the same thing with proteins. We basically this is work by Emma Tissinger in my lab. For her master's thesis, she basically looked at a knowledge graph that was built by our close collaborator, Marek Zitnik, over at the Harvard Medical School. So Prime KG, this knowledge graph where every gene is connected to diseases and phenotypes and drugs and anatomy and pathways and cellular components and biological processes. So you basically are taking a knowledge graph and you're applying a graph convolution neural network that basically, again, at every layer abstracts the representation, the Z of every one of these points based on the neighbors. And now guess what? Drugs themselves get a latent representation in that knowledge graph space, and proteins do too, and diseases and phenotypes and anatomy, every one of those gets some kind of latent representation. And now you can take protein structure and say, hey, does protein structure correlate with where different genes end up in this latent space based on this knowledge graph that knew nothing about structure? And you can learn a multimodal representation that starts translating back and forth between function and structure. AlphaFold is amazing. It solved the sequence to structure problem, but the structure to function problem is still unsolved. This is a step in that direction. And I want you guys to take another 5 steps in that direction. And we can use that to now start co-embedding protein structure and chemical structure and literature in a joint biological space and start asking questions about drug safety, about drug efficacy, about how to design new molecules and so on and so forth. Okay, so that's where the field is going. It's really the unification of all of these different aspects thanks to generative AI. Okay, here we just took this tool that I mentioned and we just threw at it every description of a gene function based on the protein data bank. And it created, this interactive landscape or I can kind of go in and ask about every gene's function. That's purely based on human language. Now you can layer in all kinds of additional data sets and go to town with it. And this is now interacting with this latent space. This is for 3 million articles of the New York Times, where you can basically now, instead of just asking, some table of these articles, you can basically see the latent embedding representations of these articles. And, this is with articles. You can do the same thing with all kinds of stuff. You can take papers and grants and patents from MIT and basically co-embed them in these latent spaces where you can basically have the grants connected to the patents, connected even to the offices in the physical space of MIT, where these are learned together. You can take every class at MIT and connect them with their prerequisite. You can basically take the dynamics over time for how knowledge changes. You can take loans and see, why are some loans refused and others accepted, and sort of look at these latent representation. You can see how people are collaborating. You can see the meaning, the minutes. Tomorrow, I'll show you a graph of your own ideas for final projects, and we'll cluster that and we'll sort of explore it based on your survey results. So that's the power of maps for both physical space navigation, for cognitive space navigation. So maps, when you go skiing, give you the landscape, the landmarks, anchor points, street names, highway names, some simplification, some abstraction, some summarization that allow you to make decisions. You can do the same thing with cognitive maps and use that to guide your thinking, to basically organize the space, organize this latent Z representation. So that's what I see as the road ahead for both this semester and for the field. That basically by understanding these multimodal, multi-layer, hierarchical, deep representations of the complex data types that we have in the field of biology, we can finally unify it. This class used to be about genomes and circuits. Now it's about proteins and images and chemicals and all of these things. Why? Because we can, because it's feasible, not because it's easy, but because it's hard and because it will be transformative for our field. Okay, so that's what we're going to study this semester. We're going to basically learn about these themes of representation learning, and we're going to, of course, dissect them in much more detail throughout a series of lectures. So again, module 1 is about expression, circuitry, regulation, epigenomes, single cell, regulatory networks, and all of that. Module 2 is about learning how to connect all this to protein structure, protein language models, geometric deep learning. Module 3 is about chemistry and therapeutics and graph neural networks. Module 4 is about images and electronic health records and evolution, comparative genomics and metabolomics. And I want you to pluck from all of these topics and design the coolest, most awesome, most ambitious final projects. Why? Because you can, because it is now finally possible. All of these things that I've wanted to do since I was your age, we can now finally do, and we can do it together. Okay? So no pressure, but you can change the world. Who's excited? Yay, awesome. All right, that's it. See you guys tomorrow for recitation. P-set 0, take a look at it today. It'll be out, finish the survey, and start thinking about crazy ideas for your projects.
Mm-hmm.